Hello students, welcome to this chapter at a glance session. Today, I am Amit and we'll be talking about cell chapter today. Cell chapter we'll be discussing in a very crux form. We'll be discussing the cell chapter in such a way that is required for right now. Means that when we are discussing this chapter, we are focusing mostly on the questions which have come up in NEET before, or we are going to discuss parts that are important. So this is not a session that talks about every minute detail, but yes, we'll try to capture the essence of the chapter so that towards the ending time when your preparations are almost done, we are going to discuss the chapter in that sense. Okay, let's start with that. Let's start with the importance of the chapter. So when it comes to cell chapter, we are trying to first get what is the weightage of the chapter. So for the weightage of the chapter, if we look into the last 10 years of this particular chapter, we'll see that around last 10 years, we'll see that there are around 5 questions on an average are appearing. If I talk about the averages, it might be around 4.5 questions. So from your biology is going to, or 5% of your entire syllabus is going to be this chapter. One thing that is pretty good about this chapter is that we have not seen, at least till now, we have not seen many tricky questions from out of it. There are one or two, we'll discuss all the questions which have appeared, but we don't find any tricky questions in this chapter till now. We get very straightforward questions, we get facts, we get scientist names, we get matchings, beautiful questions, you get high scoring chapter, easy chapter. Just to give you an idea that how important is the chapter and how uniformly you are getting the chapter, we have this. 2023, you had 4 questions, 2024, 4 questions, 2025, the last NEET have around 5 questions in it. So that is an important chapter. I have seen maximum around 6 to 7 questions, not more than that, but I have seen 5 questions around an average that are available. It's an important chapter for that matter, high scoring chapter. Let's go to the next part. So now when we know that how important the chapter is, let's go to the next section in which we can discuss the overview of the chapter. When we are discussing the overview of the chapter, we have created this particular part for you. This carries the entirety, the essence of the chapter. It contains all the important points in a single way. You can actually, if you want, you can pause at this moment, you can look at this, or it will be available at the end also. You can actually look in the presentation and you can have this and have a look of this chapter. This is something, okay, I am looking at the chapter, my last 3 days are remaining and all that. Let me have a quick revision of the chapter. Use this mind map and you are done. You have looked at around 72 to 80% of the facts of this particular part through this. Let me just go through it. I know that most of you have done most of the things already in this particular chapter. But let's go into this because, you know, smallest points, they go off. Sometimes we believe that we are comfortable in particular fact, particular point or something like that. But that particular thing gets jumbled up during the exam and create problems. So let's talk about this. Let's start with the central part of the chapter. We are having cell as a unit of life. In this, what we have done, we have broken down into 8 specific points. These are the 8 main parts of the chapter that you have to do. To be very frank, I can actually predict 2-3 questions which are coming continuously. So the first part of the chapter is about the introduction. It talks about the scientists which were involved in the discovery of the cell. It talks about that what is cell theory and all those parts. So in this, when we are discussing this part, question on Rudolf Virchow, question on tenets of the cell theory, and when it comes to the size parts of the mycoplasma and all that, they have appeared before. In this, we have deliberately added the word totipotency here because this is the part that connects to plant growth and development section and also connects to the plant tissue culture that is given in biotechnology chapter, where it talks about the totipotency nature. Remember that totipotency is directly connected to cell theory. We'll discuss it later on as more and all that. Now let's come to the first question that is almost fixed. First question that is appearing, that is from the prokaryotic cell. Every year we are seeing a question from prokaryotic cell. The problem here is that prokaryotic cell is shared with biological classification also, because Monera is there. So sometimes what happens that some questions go into Monera, some questions go into, so we have taken only core questions. We have seen only core questions, but when we are discussing prokaryotic cells, some of the points like plasmids, like diseases they cause, you will find them in biological classification. So that part is important. Connect that section to this particular part. Questions that we have seen regularly, I want you to see that the glycocalyx part, slime layer and capsule. One connection that I will build, you might have seen a diagram of nostoc that is given in biological classification. It does talk about slime layer over there, and it talks about gelatinous layer. They are talking about this. So one question, if I want to make a question, I will make it like so that glycocalyx is slime layer and all that. We'll come to that. There's one very interesting question that appeared before. I'll come to that particular question later on. Genetic material of the bacteria, nucleoid, how it is. Now, this part is better explained in the molecular section that is there. Next part, we have other structures in which we have ribosomes, inclusion bodies, flagella of motility. Flagella connects to the eukaryotic flagella. Also, very important part, both the flagella are analogous to each other. They have no similarity as the structural part, but they have functional similarity. That is important section. Let's go to the next part of this particular chapter. That is the cell boundary and the wall. I have taken up the cell membrane section and the cell wall section, and we have put up together into one single place. Again, a very important section. When we go into NCERT, we'll discuss about it. Small sections, we have taken all the smaller points and put them here. We have talked about the specific sections. Let me bring to one particular specific point that we might lose later on. Can we see here? We have talked about the plasma membrane section. And in plasma membrane section, we have written this word phospholipid bilayer, proteins integral, peripheral, and carbohydrate section. Now, this has been a repeated question in matching. This has been an important question that is appearing regularly. Active and passive transport, you might use that in zoology section where you talk about the hormone movements. That part is also covered here at this particular part. Here cell wall is the next part. Very important part in cell wall is, here can you see this? That we are talking about this algal cell wall composition of plant, cellulose, fungi, cartin, algae. A small section of a cell wall about galactons and mannans. That is also mentioned in this particular part. It is not available directly anywhere else, but cell wall of plant have galactons and mannans also. This is the part that is mentioned here. Okay, let's go to the next part. Though we have already discussed the plasma membrane and we have discussed the cell wall, we are discussing the eukaryotic cell. When we are discussing eukaryotic cell, specifically, we have to talk about the organelles and all that. So you have a big portion of the organelles that is occurring in endomembrane system. Then we have taken semi-autonomous organelles, which are mitochondria and plastids. And then we have taken up other that are the other outer or the other components also. So basically, the 5th, 6th, 7th points are actually part of the eukaryotic cell, which are given line by line. Again, small things there. Very important question. Can you see the third word from the top? That is compartmentalization. It has made last 5 years. We have seen question on compartmentalization. Let's come to the next part. We'll discuss the diagrams and all. There we'll discuss the differences between plant and animal cells. Endomembrane system, I think one of the most repeated question is endomembrane system question. It is appearing multiple times. It has appeared multiple times. When we discuss it with NCERT, we'll discuss that particular part. Very minute points are available. Just look into that specific points and just try to remember them. Next part is going to be semi-autonomous organelles, which are double membrane. But see, this word semi-autonomous, I have deliberately used it here. The reason for using it here is because NCERT doesn't use this word, semi-autonomous. But we'll see the question, a question in 2016 that appeared, which actually talks about the semi-autonomous nature. We'll come to that later on. Though remember that mitochondria and chloroplasts and plastids are better explained in other chapters. They are explained in the chapter of photosynthesis and respiration. That is the key and most important point. So you will see in summary of this chapter that they will use oxidative phosphorylation as a term. Now that oxidative phosphorylation is nothing. It is talking about the mitochondrial function. Easy sections, but at the same time, you have to connect the dots there. Very important parts. Now you talk about the other components of the cytoskeleton, in which we have ribosomes, cytoskeleton, cilia and flagella. Let's make a connection here, a very important one. See this. See cytoskeleton. In cytoskeleton, we are discussing the filaments of microtubules and microfilaments. And then we are talking about cilia, flagella, and we are talking about centrosome and centrioles. Actual reality is that cilia and flagella, centrosome and centrioles are actually made by cytoskeleton. They are made by microtubules. So any function that is given in a cilia and flagella or a centrosome and centrioles is also the function of what also? Cytoskeleton also. So let's say I've said the cytoskeleton is cytoskeleton involved in motility? Yes, it is involved in motility because it is part of cilia and flagella. Is it involved in spindle movement? Yes, it is available in spindle movement. So that small connection has to be made. Ribosomes are better explained in there are other sections which are the molecular basis of inheritance, but at the same time the dimensions of this particular part is mentioned here. At the same time, the explanation of Svedberg unit is also given here. Very important part, don't miss out on this. This is an overview of the chapter. This just give you an idea. It will start you up with the chapter. Now you have known everything. Now I know which are the places which I may remember or I might not remember. In this, the last and single most, so I talked about already this plasma membrane that it will make one question or glycocalyx make one question. One question is always and always appearing from nucleus section. When it comes to nucleus section, it acts as a single thing. Questions we have seen on nucleolus, we have seen questions on the nuclear lamina, we have seen questions on chromatin, we have seen questions on chromosomes, we have talked about the structure of the chromosome. All those questions do appear from nucleus. At least expect one question from this particular part. This is a basic outline. Go ahead, look into the outline, have a feel of it, and then we'll talk about the next part. Let's talk about the next part. Again, I have not discussed everything. Vacuoles are there, lyosomes are there, easy sections. Go through them fast. Okay, let's go to the next part. So when we're discussing this, what we have done, we have actually created certain, tables for you. Now, it is not possible to look at NCERT again and again and try to remember all the facts and all, because when it comes to matching question, statement question, factual statement questions, you will actually see these words coming up again and again. So what we have done, so now I am showing you the next 4 tables. These tables are data that is extracted from the chapter. The first table is dealing with the scientist section of it, scientist part of it. When it comes to scientist part, I have mentioned this, that one question from G. N. Ramachandran actually came in NEET. So I will mention later on the other, where the other NEET questions have appeared. These are the scientists and these are their contributions which are available. In most of them, we have added the years, but you have to add years also, because matching makes an important question in this particular part. So we have first Mr. G. N. Ramachandran. Main important points: Madras School of Conformation Analysis and Biopolymers that he established, and Ramachandran plot, very important point. One small caveat in Ramachandran, I'll come to that later on. It's a very interesting point actually. Let's go come to the next one. Who saw the first cell? Anton von Leeuwenhoek. I'll give you one thing. In most of your cases, you might have done that who discovered the cell, and Robert Hooke is the name that comes up. Actually, NCERT doesn't use the word Robert Hooke. It is not there. So NCERT directly starts with a living cell. When it comes to Robert Hooke, we talk about cell wall and a dead cell, but we don't talk about a living cell. So that is Anton von Leeuwenhoek, Robert Brown about discovery of nucleus, Matthias Schleiden and Theodor Schwann. I know that you are all students of this year. Probably you have made this change already. Let me tell you about that change also. See, this German botanist is written here, and here now it is German zoologist. Earlier it used to be British zoologist. Please don't make that mistake. It is now German zoologist. Important particular point. There's a small thing about Theodor Schwann also. We'll discuss that. A small confusion that NCERT creates. Discuss that part also. It have potential to make great questions. Now, Rudolf Virchow. If you see last 5 years, Rudolf Virchow have appeared in last few years, in which who modified the cell theory, who gave hominis cellula, e cellula. This keep on appearing. Even in matching, Rudolf Virchow appeared so many times. We have Singer and Nicholson, who gave fluid mosaic model or quasi-fluid nature of the plasma membrane. Then you have Camillo Golgi and George Palade, which have worked on Golgi bodies and talked about the ribosomes. That's why ribosomes are also called Palade particles. And towards the end, the nucleus, you will find Fleming, who talks about the chromatin section, and the term chromatin is coined by him. It's just look at the table, revision done. Next one. Second table, what we have created here, we have taken all the organelles. We have broken down all the important organelles we have taken. We have broken them down into their components, and we have given description and function of those components also, because questions. That's all. I'll give you an example. Let's say a question, generally nucleus might not be the question, but nucleolus might be a question. So what is the function of the nucleolus? What is the particular function of chromatin, function of cristae? These questions keep on appearing. We have put them in together in this particular part. You can, if you want, you can pause the video, look at it, just have a quick revision of it, have a beautiful feel of it, understand what NCERT want to say, because these will be single statement question or match the following question. Easy questions. Let's talk about this. So we are talking about cell membrane, we are talking about phospholipid layer, the membrane proteins, and cholesterol. Now, cholesterol is specific because cholesterol as such is mentioned in biomolecules. It is also mentioned here in the diagram, but question about cholesterol as such are not there. So the stability part of the cholesterol is not discussed. So that is, we have mentioned that also here. Next part is going to be nucleus. In nucleus, you have nuclear envelope, nuclear pores, nucleolus, and chromatin. Now let's talk about a small question here. Now see, there is a small caveat that is given in nuclear envelope, that the perinuclear space is 10 to 50 nanometer. Now there is an older question which I believe is a good question. So now you know that the distance of the perinuclear space, the distance between the two membrane is around 10 to 50 nanometer. But let's say I can reframe this question a different way. I'll say that what is the depth of a nuclear pore? So nuclear pore is going through the membrane. So that is why nuclear pore will have the same depth as the what? As the nuclear envelope. So this is an important section. Then you have nucleolus. Nucleolus have made question in last 2 years where they talked about that what is the main function, that is RNA synthesis and all that. rRNA synthesis basically. Now I can, if I want to make a complicated question, I will take this question and I connect it to the molecular biology section, in which I will say that where is rRNA synthesis happening. So I can give you nucleus, but I can also give you nucleoplasm or nucleolus, all those words also to make you confused about this particular part. Let's talk about the next part, mitochondria and chloroplast. They have to be done together. Good questions are coming from them. So about mitochondria, the cristae, the enfoldings. Remember that it resounds with a word called cisternae that are actually found in ER and Golgi apparatus. Nothing to do here. Sometimes students read cisternae and cristae differently. Don't do that. Important section. Next one, matrix. Now how matrix connect? Matrix can be a part which can be connected to Krebs cycle in respiration chapter. So important part is there. That which parts? So are all the enzymes of Krebs cycle in matrix? No, they are not. There is succinate dehydrogenase which is actually present on the membrane also. So remember that inner membrane is a site for ETS. Matrix is a site of Krebs cycle. Good questions, but respiration questions. Next one's a chloroplast about thylakoid stroma. We mentioned flattened sac-like structure, like a coin, like a stack of coin, they are being there. Then you have endoplasmic reticulum, in which rough ER, smooth ER are present. Their functions are separately given. When we discuss the NCERT, we'll discuss that particular part. Then you have Golgi apparatus. Cisternae word is given in a Golgi apparatus. They have talked about cisternae, tubules and vesicles also. But I have specifically mentioned cisternae because question recently appeared where they talk about which is the forming phase and which is the maturing phase. Next one is cilia and flagella. Exoneme is a very good question. When we come to the diagram, I'll discuss the exoneme. Next one's about basal body. Now basal body is a good question. Why it's a good question? When it comes to centriole. Centriole have a 9+0 microtubular arrangement. 9+0. Flagella have 9+2. Cilia have 9+2. Now I can ask a question to you. What is the microtubular organization for the basal body of the flagella or basal body of the cilia? Remember that NCERT have given that centrosomes or centrioles are the ones which are actually modified into them. So if I talk about flagella, answer is 9+2. If I talk about cilia, answer is 9+2. If I talk about basal body of flagella, answer is 9+0, not 9+2. Good question there. We have talked about the hub and spoke model of the centriole also, and you can have other sections which have cartwheel-like model also mentioned when it comes to the cilia and flagella section. let's go to the next table. You see the questions above. We have seen question in 2025, 24 actually twice we have seen from this table, we have seen the question 2021, 2015 and 2015 July, because there were 2 exams over there at that particular time. Let's go to the next part. Next part is about dimensions. I don't have to tell all those things, but there are a lot of dimension questions. There are also mycoplasma, what is the size? What is the size of a bacteria? Now see, this 3 to 5 is the length. Typical bacteria is around 1 to 2. That is the basic diameter of the bacteria. Human cell, mitochondria, chloroplast, Golgi cisternae, nuclear pore, DNA. See one question here. Specifically, I want to talk about mitochondria and chloroplast. See, mitochondria length is 5 to 10. That's quite varying. And then width is also there. There's a line given in NCERT according to this. It says that a chloroplast like structure, cylindrical structure, but it can be oval structure. It can be egg-like structure also. So remember that the line that when it comes to plastids, the number can vary cell to cell, the shape can vary and size can vary. In NCERT, the number part is specifically mentioned for mitochondria, but they have not mentioned anything about the shape and size. But remember that because these ranges are mentioned, there is a part here where size and shape even for mitochondria can be different. So if I write it in one statement, that do mitochondria have or can have different shapes and sizes? Yes, it can have different shapes and sizes also depending on where it is present. So again, we have taken all that particular part. Dimensions are there. Just pause the video for 1 minute, go ahead and you can go into everything. We have taken the key important part also, or key important point to remember about it also, and nothing much is required in this. Now you have the dimension part also done. Let's talk about the last part of this particular section. That is going to be the function sections. Now remember, there can be large number of functions which are available when it comes to the organelles. But when it comes to this section, stick to NCERT functions. If I see from 2018 onwards, we have seen that the functions of organelles are coming in context of NCERT also. So I have taken deliberately those lines which are available in the NCERT and put up all the functions. So yes, you might think there are other functions also available, but my idea right now is that I'll tell you this, stick to NCERT functions. If one function is mentioned in NCERT, focus on that particular function, go to that function only. So I don't want to spell all the functions to you, but they are all there. Just have a pause of this particular video. Go ahead, look up at all of them. All the things are mentioned, which are transition sahi lagega na? Okay, so till here we have discussed the parts or the contents which are given in NCERT. We'll discuss the diagrams that still has to be there. I have to discuss the hotspots of the NEET questions also, that I'll discuss. But before that, let's do that. Let me take you to NCERT itself and see, is there a pattern available when it comes to NEET questions? If there is a pattern available, we'll find that pattern and we can actually mark those things. So at this particular juncture, I want you to take out your NCERT, have that NCERT in front of you and see where the markings are. You will actually see that NEET is giving some questions again and again every year. So whatever happens, I am not leaving those lines. So let's go to that particular part. Let me open the other document for you. So let me take you there. So this is the, just 1 minute. So we are going into the NCERT. I am starting from the unit part of the chapter. I am not starting just from the chapter. We are starting from the unit part. So I am not explaining NCERT here. Main mode is, let us mark the NCERT. Let's see where the questions have appeared. That make them a high incidence question. Means those questions can appear again also. So let's come to that. So I'm going page by page, one by one with everything, and let's see what is there. Let me start somewhere here. Can you see this? It is the question last year. NEET 2025, you have the question, physicochemical approach about reductionist biology. Important question appeared. Please mark that up. That means it gives you an idea. It doesn't tell you that this question is going to repeat. It actually tells you that when it comes to all the chapters, cell and all those chapters and all that, the unit descriptions, this is the not a description of the chapter, it is the description of the unit. From there we can have some questions as well. So that is what is happening here. Let's go to the next part. Let's see what is there in the next page. G.N. Ramachandran is this next part. The scientist that is available here. 2019, we have seen question on G.N. Ramachandran. Let me make a point here also. So that Ramachandran the question was given on what was his contribution. But see one thing here. People think that this is about Ramachandran, but can you see one thing? That except for Dr. Ramachandran, we have a small idea about Linus Pauling also here. So now that is a interesting point. It is about this, but if I want to give you a matching and if I want to give you a statement-based question, I can give you about Linus Pauling as well. Let's move fast. Let's go to the next section. Let's see how it is. Okay, again, keep your NCERTs ready. We are going one by one. We are looking into everything. Let's see what is happening. I already discussed this. Anton von Leeuwenhoek saw the first cell. So many matchings. Robert Brown, they will take the matchings. They will take scientists from other chapters also, like chapters from biotechnology, scientists from biotechnology or scientists from other sections. And they will have matching questions from them. Very common question, but don't forget to mention this particular part. Here, remember that Matthias Leden 1838 and 1839 is given for Schwann. Now come to this. I want to show you something important part. Let's see this. So again, Rudolf Virchow, already mentioned here. If you see Rudolf Virchow, this is 1855. Omne Cellula Cellula has come up in matching in 2019. It has appeared in a question before as well. Now see a specific point. When we discuss it here, German zoologist already discussed with you that why it is available. If we see certain small things, these are the only 2 points given in NCERT when it comes to cell theory. There are no other points. The virus being exceptions is also not directly mentioned as such. Important section. Let's go to the next part. Let's go to the next part of overview of the cell. Can we go this? 2010, very old question, but again, question appeared. Main arena of cellular activity, single statement question, make a good question out there. On the same page, you can have NEET 2022 question where mycoplasma is the smallest cell that have appeared. You all know this. Biological classification also, you have the same thing available. So you do it there actually. Let's go to the next part. Okay, these are the diagrams. Now, see again, diagrams will come later on. So let's discuss diagram there only. Let's see the next part. In the next section, we have description of the prokaryotic cell. PPLO has come up as question also. And at the same time, the sizes, the bacillus, coccus, vibrio and spirillum has been also asked in match the following questions, or multiple matching questions have appeared around them before as well. Let's go to this. I am using the latest version of the NCERT. If you are somebody who is still using the older version, I'll suggest have the latest version with you because it have some small changes also. NEET July 2015, the one that got cancelled. We have this, the semi-fluid matrix filling the cell. So they talk about this, the nature of the nuclear membrane and all that. Very basic thing, but it is mentioned an important part about the bacteria where they talk about there's no nucleus and all that. Easy portion, but part of the question. Let's go to the next part. We are discussing the next section. Yes, already discussed glycocalyx with you. Now see, this NEET Karnataka 2013, where NEET was adopted in some states, it was not adopted in other states, or some states were having their own NEET at that particular times. Let's not go there. Glycocalyx, their composition. The question actually came in 2017 on the sticky nature. The next part. Let's talk about this when we are discussing glycocalyx. I already discussed the glycocalyx before. Though the glycocalyx question, can you see this? In 2017 NEET question came on the sticky nature. Now, technically glycocalyx sticky nature is not mentioned here. But you have done something called Griffith experiment that is present in molecular biology. So that molecular biology section connects to here. That they're talking about the glycocalyx. Glycocalyx is one version of glycocalyx is capsule, and capsule have which nature? Is sticky nature, which make it or which give it smooth colony. So that is the part. So let's go to the next one. We have in 2012 question. This is a good question, that what are the similarities between bacteria or the monorans and the eukaryotes? So the key important part is that they have similarity in their plasma membrane. Let's go to there now. 2024, very recent question. The question was about the mesosomes. Question was about the functional aspect of the mesosomes. They said that which of the following is true about mesosomes, or the statement is about the vesicular nature of the mesosomes. So that is there. So they said that mesosomes can be what? So they can be vesicular or not? Yes, they can be vesicular. So that was the question. Tubules, lamellae, they are also the words that are particularly used here. Now, if you see this, so I'm starting, this is the new blue section. This is the 2025 question that we are discussing. So the question in 2025 actually came around the functional aspect of the mesosomes, in which they have given certain functions and they have asked that which of the following is the function of the meosome and all, and their respiration, secretion, increase in surface area of the plasma membrane, DNA replication, distribution to daughter cells, and all those things are given in this. Very straightforward question appeared, easy question appeared, important question 2025 mesosomes. We are seeing 2 questions in last 10 years, that means it is an important section. Pilae, fimbriae have been appearing in the matchings and all that, but again they are easier section. I'm going for the statement based questions here mostly. Inclusion bodies also, it appeared in NEET 2015, and you have 2020 NEET also, where we have inclusion bodies, reserve material part have appeared, that are the membrane bound or not. Very structure, very singular question that appeared, easy question that appeared from this particular part. Please mark that section. Let's go to the next section. Okay, so here you have diagrams of both animal cell and the plant cell. When it comes to the diagram at the end, I will discuss the diagrams one in a very fast way. Let's go to the next section, in which we are talking about the next part, that is the cell membrane. Cell membrane has made question. Can you see that? NEET 2025, 2012, 2010. Somewhere here and there, some statement questions also have appeared, but the phospholipid nature, the bilayer nature of it, the position of the, if I see 2025 question, you have that what is the position of the lipid, that was the question was around that, in which that who is towards inside and who is towards outside, those kind of questions have appeared. Important part about the plasma membrane. We are coming to the diagram together, we'll discuss at the end. Just give me some time, we are going to go and we are going to reach there. Let's go to the next part. Next part is about the fluid mosaic model, in which year it has appeared. In the fact section also discovered, but the question have appeared in 2012 when it comes to this particular part, that who gave this model, when the model was given. Passive and active transport in 2012, again 2 questions in one paper appeared that year. Again, when it comes to cell wall, there 2010, you have 2 questions from the cell wall part. I was talking about something called galactons and mannans. Can you check that? That you have galactons and mannans available here. When we discuss algae cell wall, we discuss cellulose, polysulfate esters, we talk about algins and all that. But remember that galactons and mannans are also mentioned in this part of the NCERT. So this is there. Let's go to the next section. Then you have cell wall, middle lamella may be transferred by plasmodesmata. Plasmodesmata have made questions before. This is how it is. Please keep on marking your NCERT. Very, very important section. So if you are marking the NCERT, let's go into much more important section. This diagram have appeared before in one of the previous papers, and very, very important section. This line which have endomembrane system, who is involved in endomembrane system and who is not involved in endomembrane system, have appeared before. Please mark this thing important. 2023, 21 means in recent papers, the paper makers have taken a liking to this particular line, important line. Let's go one by one. Let's just mark all these things and all. Now come to the mother of all questions. So this, see, RER, SER, this is such a repeated question. 2022, 2018, 15. I've written 13 2 times. The reason for that is that because in 2013 paper it actually appeared twice. So what are the functions of RER? What are the functions they are doing? What are the functions of a smooth ER? What are the functions or what make and all? Some way or other, we expect a question out of it. Very, very important section. Go through it beautifully. Do it. Try to remember it. Okay. Diagrams I'll come towards the end. This particular part, the flat disk-like structure about the cisternae that we are talking about, that cisternae have appeared in a match of the following question, 2021, 2015. We have questions from this particular section as well. Cis and transphase, this particular part, cis and transphase have made questions before. So this, if I am a paper setter, I want to give question from this. I want to give question from cis and trans nature, how the maturity happens and all those things. Important section. Important lines. I'll say Golgi apparatus ER, they will make a question. They make a question almost every week. Let's see this. We are just into Golgi apparatus. And can you see this? In Golgi apparatus, it is important site of formation of glycoproteins and glycolipids. The question on glycosylation, glycosidation have appeared. Can you see NEET, October 2020, September 2020, 2019, twice, 2013 and 2011 question have appeared. So all in all, endomembrane system, and in an endomembrane system, when it comes to ER and Golgi apparatus, we are going to see questions again and again from this section. You are getting at least one question from this section. Let's go to lysosome section, 2019, 2016, phase 2, and then you have 2012. You have these questions from them. So they talk about that what kind of molecules they can preserve, what kind of molecules they can digest. So lipase, proteins, carbohydrates are mentioned here. They can actually digest almost every biomolecule that is available that you have studied in biomolecule chapter. They can digest that particular thing. But lysosomes have made questions before. Let's go to the next one. Vacuoles, NEET 2019, 2016. A question that is slightly off. 2016 I have taken a question because slightly off NCERT question. Question was about which of the following is a water soluble pigment, and the answer was anthocyanin. So anthocyanin is generally sometime a vestigial pigment. It is not used that much, but it gets saved, or it is a water soluble pigment which actually get dissolved into the water and is now found in vacuole. Important question, 2016 out of NCERT question. Let's go to the next part. Coital vacuole have appeared. If you see the tables, we have taken all the major facts and already present there. Let's go to mitochondria section. So I was talking about mother of all questions. Let's talk about father of all questions. So structure, this particular diagram have appeared in before in 2011. And then come to this. The inner membrane, infoldings, cristae, ATP synthesis, what kind of ribosomes are present in the matrix. Important section. NEET 2025, 21, 19, 2017, 2016, 15, 14. You have so many questions appearing. So again, a question that will appear. A problem with these questions is that many of us might think that, you know, this is not the question from this part. It is actually a question probably from respiration chapter. But these lines are the ones which have made questions. They have talked about this. Here I want to mention one thing. So when I discuss this, here it will go. Can you see this? This one, this. Can you see there they use the word single circular DNA molecule. Many students think that they are talking about a single stranded nature. They are not talking about single stranded nature. They are actually talking about one molecule of DNA. They are not talking about the single stranded nature. Important portion, small thing, small point, make a note and move forward. Let's go to the next part about the plastids and all. In plastids, they have talked about that carotenoids have, chromoplasts have what? Chloroplasts have what? What are the important things they are available? Leucoplasts are made up of which one? Elaioplasts and myeloplasts, they have appeared in questions before. 2013 is one of the question where the fat soluble carotenoid pigment part was discussed once. Let's go to the next part. Again, let's mark the NCERT. Let's see where the things are important and all. Okay. This line, like mitochondria and chloroplasts, are also double membrane bound. Of the two of the inner chloroplast membrane is relatively less permeable. Very important line. 2024. A question that bamboozled so many students. Students had problem with this question. Towards the end, let me discuss that question also with you. That what was the crux of that question, why students got problem with it. We have mentioned that question. I am going to discuss in a separate segment altogether. And now the next part, in which the stroma, thylakoid arrangement of these sacs and all, that is given 2021, 15, 2015 again, then 2014, 2012, repeated question. So mitochondria. So one part of the question is endomembrane system going to appear. Next part of the system is going to be the chloroplast and mitochondria. You will see a question from there. Out of the 5 questions, 2 I would see from here. Here you have other part in which you have chlorophyll pigments, where they are present, thylakoids, all those parts are there. Ribosomes are important question, but ribosomes you do much better in molecular biology. But yes, remember one part that many students don't see, though we have already made the table around and all. So this 70S ribosome and 70S ribosome, 80S ribosomes are mentioned, very specific things, small subunit, large subunit. This diagram can also appear. NEET 2025 actually made a question around it in which they talked about that what are the subunit type of question which have appeared in last year. Then you have cytoskeleton sections. Cytoskeleton I've already discussed with you already. They are talking about that particular sense only, because cytoskeleton have overlapping functions with cilia, flagella, centriole and all those. That's why make it a very important question. Part the mark from green 2010 made a question. In NEET 2023, 2016, 2014 we have these questions, or questions from the functional section, that is mechanical support, motility, maintenance of the shape of the cell, have appeared before in the questions. Let's go to the next part. Again, important diagrams. We'll discuss the diagrams at the end. Now you have cilia and flagella part. 2025 question. Question was about the ciliar structures and all. Easy question. They talk about the movement part and all, that what is the how the cilia work and all, how they around. Important question 2025. But see one thing. In a recent papers, you have only NCERT centric questions. You are seeing these questions which are taken from NCERT lines, modulated NCERT lines, but they are NCERT lines which are making this question. Let's go to the next part. So axoneme I have not mentioned here, but axoneme have appeared in many of the matchings before. Then cilium flagellum, and I have already talked about the relationship of them with the centriole. That is the part that made question 2014 and 2012. See, these are the areas that I have marked. They are subjective in nature. There might be other questions from in other statements also. But remember this is just to tell, just to convey the importance of these lines, small NCERT lines, which are making so many questions over the time. Let's talk about the centrosome. So I have not marked anything in centrosome, but that doesn't mean that it's something that is irrelevant. Now I'll give you this example. See this cartwheel part. This has appeared before. Now let's say the line that has not appeared. Let's imagine this line. They are surrounded by amorphous pericentriolar materials. Now if I want to make a new question, I will go with this. So important lines, don't forget any line. Only thing is that till now we have not seen many questions from this particular part. Yes, but the lines have appeared, or they have appeared in matchings and all. Let's go to the next section. So we are going towards the ending of the chapter almost. We have now reached the nucleus. In nucleus part, about the chromatin, Fleming have come up in matchings so many times. NEET 2024, 2015, we have seen the question. 2015, we have talked about the chromatin fibers, nuclear matrix, what are the composition, those particular part. NEET 2021, about the perinuclear space, this dimension have come up in a question. In 2015 cancel version of the NEET, we have seen the question on this, where the multiple statement based questions have appeared in 2015 particular part and 2011. As you go down, nucleoli or nucleolus has been an important question in nucleus, in which you can see NEET 2019, 2018, 2012, we have seen questions from this particular part. What is the function? Function is about ribosomal RNA synthesis. Is it membrane bound or not membrane bound? Those kind of questions have appeared before in this particular section. Now I'm going towards the end game. We are going towards the ending part of the chapter, that is about the chromosomes. Chromosome is also somehow a mandatory question. So many questions have been appearing on the chromosome part. So what is happening there? So when we discuss this chromosome, what they are made up of and all, there's a small interesting point that is hidden here with the structure of chromatin. I'll come to that towards the end. I'll come to that very interesting part. Then you see centromere section, the position of centromere, telocentric, ectocentric, metacentric, and some metacentric, those have appeared. I have marked everything. Last year matching appeared, before that matching was 2021, 2015, 2000. So this diagram have appeared before and all which have appeared. Though one point I specifically want to make, see the word telocentric. In the word telocentric, we say terminal centromere. So many students believe that this is a telocentric condition, though the text and the diagrams are not matching with the particular thing. NCERT nowhere it mentioned that this is a telocentric chromosome. So this is one thing, make a note of it. Telocentric diagram is not mentioned in NCERT technically. Let's go to the next part. Let me go to the last part. Let me take you to the microbodies. I just mentioned the microbodies because this is one single line towards the end and many students have trouble with it. Remember that microbodies you have not done directly, but peroxisomes have been discussed in photorespiration part of the photosynthesis. Then glyoxysomes and spherosomes are not discussed, but please do have a look at the peroxisomes. They have made questions before. Let me take you to the summary section. And when I go to summary, I will want to take you to this line. I told you that there's an interesting point about endoplasmic reticulum that I want to discuss. That is actually this point. When it comes to endoplasmic reticulum, tubules and cisternae. Now see this first. Tubules and cisternae. Cisternae word is used in the main text or in the main part of the NCERT along with Golgi. But here also they are using what? They are using the word cisternae along with it. That is first. Now, as we go into the functions part, ER helps in the transport of the substances, synthesis of proteins, lipoproteins and glycogen. Now, this word glycogen is not there in the main text. So, when it comes to this, so glycogen is also mentioned. Glycogen synthesis done by SER, important question. Don't forget to make a mark of it. I marked one more line here where mitochondria help in oxidative phosphorylation word is mentioned, important part. Though you will have a better understanding in the respiration section, but important section, generation of ATP is also there. So please read summaries of all the chapters. Here also make a point that you are actually talking about the summary of this particular chapter. This is all when it comes to NCERT lines. Hope you have marked the lines. Wherever you feel that, okay, I am marking, pause the video and mark the lines over there. Okay. Let's come to the next part. Let me take you back to the presentation. We have discussed most of it, the things we wanted to discuss. But because we are doing this, let's go through the diagrams once again fast. Though we have seen all the diagrams, but if is there any nuance that is available with the diagram? Is there any issue that is available with the NCERT diagrams? So we are going to the presentation part. Let's go into the diagrams actually. So let me take you to the diagram part. So again, this is just reminding the diagrams that these diagrams exist. Make a point that these diagrams are burned in your head because these diagrams are appearing. We don't know which diagram is going to appear. New diagrams have been taken up by NEET also, but they are not repeating the diagrams as such, but they're using the diagrams and all. Please just have a mental note of them. RBC, that is round and biconcave, extra information is given here. White blood cells, amoeboid, extra information is given. Columnar epithelial, long and narrow. I can see a very good match the following question appearing here. Nerve branch, branch and long, tracheid that is elongated, and mesophyll cells. Now let's say, let's have a play with the small question. If you're looking at this mesophyll cells, if I give parenchyma, will it be wrong? No, it will not be wrong. It will be correct only. So again, if I want to make it a complicated question, I can. So I should be remembering each and every diagram. Make this mental note that these diagrams do exist in NCERT. Let's go to the next one after this. Okay. Next one, I have mentioned the AIPMD. This diagram as it is came up. They asked that what is this? They have given this particular value. Here you have eukaryotic cell given 10 to 20 micrometers, typical bacteria 1 to 2 micrometers, PPLO 0.1 micrometers, and viruses are given as 0.02 to 0.2. Now, if I want to give you a complicated question, what I can do, I can convert this micrometer into nanometers and I can give you that particular question. So, in the 10 to I can make that micrometer into nanometer and I can multiply by 1000 and I can get that particular question. So, that should not happen to you. Let's go to the next one. Let's go, important diagram. Hard diagram to come in NEET, but I can give you a version of this diagram. Make an important note that this is about what? This is cholesterol. Cholesterol is mentioned here. Cholesterol nowhere else is mentioned in NCERT, though in your table that we mentioned earlier, it is available. Cholesterol is mentioned. Now, one question on this. Now, see, integral proteins are mentioned, peripheral proteins are mentioned. Now integral, peripheral, those parts are there. But now if I give a statement to you that on the basis of position of proteins, they are integral proteins or they are peripheral proteins, that will be a wrong one. That will actually be wrong, because NCERT, what it uses? What is the word that NCERT uses with it? NCERT actually uses, it talks about ease of extraction. So remember that you have ease of extraction as a criteria, not exactly the position as a criteria. See one more thing, one more nuance in this diagram. See, these are sugars, but this is a protein. The problem is that many times when a diagram is appearing, they will make diagram something like this, and you might think that this is also a sugar. This is not a sugar. This thing is actually a protein. People remember this. It's a big bulbous structure is protein. They remember other parts as proteins and all, but they don't remember these parts as proteins. So important one, remember that this is a protein, this is not a sugar. Important point. Let's go to the next part. Let's see what is in the next section. Again, I have taken all the diagrams. So this is a diagram of ribosome, you have mitochondria, and you have chloroplast. Now, it's a three-dimensional diagram, hard to give because in NEET papers, we don't have color diagrams available. So at times it is hard. So when you are trying to remember this, do one thing, remember this sequence also. Because sometimes, you know, that this line might not be very clearly visible to you in the diagram when the questions are coming up. So do that when you are trying to remember the sequence. This is outer membrane, this is inner membrane, this is intermembrane space, matrix, crista, because they are important points there. Similar thing here, again a three-dimensional diagram that is given for outer membrane, inner membrane, granum, thylakoid, stroma, lamellae, have a mental note of them. I already talked about the ribosome section, important section. Let's go to the next section. We have taken two diagrams, one is of Golgi and one is of the endoplasmic reticulum. This brings me to a question. So there's a line given in nucleus part that the outer membrane of the nucleus is continuous with the endoplasmic reticulum. Who is outside? It is actually RER which is continuous with. Now if I ask you a question that are ribosomes present on the outer membrane of the nucleus, the answer will be yes, because outer membrane is continuous with the RER. That's why ribosomes are also present on what? On the outer membrane of the nucleus as well. See, in this question, in this particular part, this particular diagram, people start to make cis and trans. Issue is this, NCERT doesn't mention where is cis, where is trans. If in diagram it is mentioned that this side is cis, you can mention this side is trans. If they mention this side cis, this side trans. They have not followed the concave convex diagram here, and that creates an issue for many of the students. Have a mental note of the diagram. Let's go to the next part. Let's go to the next diagram. Next is, I've taken this diagram of the central of the cilia and flagella. Now see a small thing. Can you see something somewhat here? See this? If you see the line of the plasma membrane is going inside. Actually, the outer part, that is the green section, this section, this is plasma membrane. This is a small part. You see minutely, then only you will see. So the inner part is not plasma membrane. There's actually the green section that is around it, that is actually the plasma membrane. Rest, everything that you are seeing inside is actually exoneme. That is how it is. This is how electron micrograph that is present, important one. I can give you this also in a NEET diagram. It's easily printable. It can be given as, and you will have problem discerning what the diagram is of. And then you have next section. In this, we have taken, I've deliberately taken this particular part, 2024, 2022, a matching 2021, question on positions of metacentric, submetacentric. All those questions are appearing. Important question, kinetochores are mentioned in NCERT like this. So remember that they are present on both the sides. There is no one kinetochore. There are actually kinetochores present on the either side of the centromere. This is again a diagram. Electron micrograph is present there. So they will give you this particular diagram and they will ask you to identify things in this. Stop, look at the diagrams and make a mental note that I have seen each and everything. Let me bring one more last thing in this. See this? Here it is given as satellite, and here it is given as secondary constriction. Now the issue is this, that this creates a problem. If this side is satellite, how this side is secondary constriction? Actually, secondary constriction is actually this part. You see this small section that is towards inside? This is supposed to be the secondary constriction. So again, second part where NCERT is slightly off, and it might save you some marks in NEET. That is, I think all the diagrams have been covered here. Last set of the diagram will be the two. One is the animal cell and one is the plant cell. Now see one thing. Let me mark specific things. See microvilli. Microvilli you do in digestion. Microvilli are done in the esophagus also. But microvilli part is not mentioned anywhere. What is the function of microvilli? That is not mentioned. But in the diagram, microvilli is there. But at the same time, in the plant cell diagram, there is no microvilli. So first make note of that. Then again, a vacuole part is there. There is no vacuole present in this. Now this creates a trouble. So contractile vacuoles and other kind of vacuoles can be present in the animal cell, but according to the diagram, vacuole are not there. See one more thing. Just see here, peroxisome are present in both of them. So because peroxisome are mentioned in NCERT along with the photosynthesis and photorespiration, many students don't think that peroxisomes are present in animal cells, but they are present in animal cells also. So key differences are these only, but they do exist, have this particular thing available. See one more last thing. See this cell wall is there, this cell wall. Now thing is, and now let me find middle lamella. See this is middle lamella. So if I see minutely in this, remember this is one cell. This is actually cell wall of another cell. That is why the middle space is what? Middle space is middle lamella. So remember that. Even these parts which you see on that upper part, this section, this side, here, this side, they are actually also what? They are also actually middle lamella. And these red dots that you are seeing here, they are actually plasmodesmata. So I hope we have a very quick revision. We just had a very quick revision of the cell, and now you have an idea what to remember and what not to remember.